ribonucleic acid. RNA is called ribonucleic acid. So I have already told you that we were doing nucleotide. So RNA and DNA both have nucleotide in their suffix, right? DNA stands for deoxyribonucleic acid. So nucleic acid is suffix with DNA. And RNA is like ribonucleic acid, means this is also, there is also a suffix that is nucleic acid. So collectively, both RNA and DNA are known as the nucleic acid. Here we are talking about the RNA. So RNA is called ribonucleic acid. RNA is single-stranded structure, fine. And DNA, we understood that this is the double-stranded structure. Length may vary from RNA to RNA. So there may be 70 to 12,000 nucleotide in a single RNA. But we have already studied that in a DNA, normally in DNA, there are only 10 nucleotide base pair, right? So this is single-stranded. RNA is the monomer of RNA, or the basic unit of RNA is also nucleotide, the same like DNA. Here again, we have understood in the last class that nucleotide are made up of three things: pentose sugar, nitrogen base, and phosphate, right? Pentose sugar, here in RNA, the pentose sugar is, that is ribose sugar, right? In DNA, it was deoxyribose sugar. Here in RNA, it is ribose sugar. Fine, clear? And again, if we talk about the nitrogenous base, the nitrogenous bases which are found. So again, here also the four kind of nitrogenous bases are found. So in RNA, the nitrogenous bases are A, C, AUGC, right? So AUGC. Here, focus over here. In DNA, we have studied. So then DNA, the nucleotide were in nitrogenous bases were in DNA. The nitrogenous bases were, they were 80 GC. But you know, here you can see that thymine is not present. So in place of thymine, uracil is found. So here the combination will be AUGC. Rest of the things will be same. There will be double bond. So in DNA, thymine is found, while in place of thymine in RNA, uracil is found. That is the difference. Thymine is not found in the RNA, and the pairing will be same. Again, I have written over in place of thymine, uracil is found as a nitrogenous basis in RNA. Phosphate will be same, phosphoric acid, S3PO4, right? Till date, we have discovered 20 different type of the RNA, right? So we can write it down over here. Till date, 20 different type of RNA of RNA discovered, but normally we study there are three type of RNA, right? Messenger RNA, ribosomal RNA, transfer RNA. So mRNA, rRNA, tRNA. mRNA is the largest one, and tRNA is the smallest one, right? So that is all about the RNA. Is that clear?
Yes, sir.
So did you note it down?
In just 1 minute, sir.
Yeah. So note down the RNA as well.
Done, sir.
Okay. So now let's start the most interesting part, that is the enzyme, and one of the most important part of it. Right. Hope it should be clear to all of you. RNA here, I have represented with my own handwritten, right? So you can directly copy paste from here, right? You can directly take the content from here. I have already filtered the content. I will read it out and I will make you understand. that what exactly I want to say, right? Enzyme. Everything is there in DL and it's very explicit over there. So what enzymes are? This is important. I have written as a notes because in your NCRT it's not in proper manner or a proper way, and it won't be you won't be able to write it clearly easily, right? So if you get the question, and enzymes are very important. Definitely you will get the question from this. Definitely you will get the question. So what exactly enzymes are? Enzymes are commonly proteinaceous substance. So there is a statement: all enzymes are protein, but all proteins are not enzymes. That is true. All enzymes are made up of protein, but all proteins are not enzyme. Why? Because sometime protein work as a hormone, sometime protein work as an enzyme, sometime protein work as a structural component. For sake of example, our skin is made up of protein, that is a keratin, right? It's a type of protein. So that is protein, and sometimes it is found in a different component. Like milk have a protein called casein protein that give white color to the milk, right? So that is also a different kind of structural protein. So protein have a various role, but all enzymes are made up of protein. This statement is true. So enzymes are commonly proteinaceous substances which are capable of catalyzing chemical reaction of biochemical origin without themselves undergoing any change, right? So what is the catalysis? Can anybody tell me what is the catalysis? Martin, what is catalysis?
They speed up the rate of reaction.
Exactly. They speed up the rate of reaction. So, but they did not get consumed in that reaction. Means they are recyclable. They are reusable, right? So that is the definition of enzyme. Chemical reactions, biological origin, without themselves undergoing any change. Therefore, they are called biocatalysts. So that's why the enzyme is called biocatalyst. I have underlined over. So if you get the question why the enzymes are called biocatalysts, you have the explanation over here. Next. The term enzyme was coined by a scientist. The name of the scientist is Kuhne in 1878 for catalyzing active substances. Previously, it was called ferments. So enzyme term was coined later after the discovery of enzyme. So Kuhne given the enzyme term. Before Kuhne given this enzyme term, it used to be, or they used to be called ferments. We used to call them ferments. So this term arises from fermentation. Enzyme were actually found out by Buchner in 1897. So finally, like 1897, and the characteristic of enzyme was defined, and they got to know, yes, these are the enzymes, right? So Buchner given this concept that they are the enzyme and what is their function. With the accidental discovery that fermentation of sugar is not only cause of living yeast but also their extract, right? So this was an accidental discovery, and they discovered it in ferments, means in fermentation process. Buchner in 1903 isolated the first enzyme. He was awarded with the Nobel Prize for the same year, 1903, right? So Buchner again, who given the term, Buchner again discovered first enzyme, and that this was he got the Nobel Prize for the same. Several enzymes of medical and chemical importance are now available in the market. Rennet tablet. So, what is the function of rennet tablet? Anyone know rennet tablet? Okay, let me check your uh, general knowledge. What is the function of renin? Renin is a enzyme. What is the function of renin? I am going to give you a clue. Renin is that enzyme which is secreted in young children, in human, and when we grew up, the secretion of renin stopped. Like you and me, my like you and me will not have the secretion of renin, right? We are not secreting renin in our side arrows. Anyone of, come on.
What was the question?
What is the function of renin enzyme?
What enzymes? I couldn't hear.
What is the function of enzyme named known renin? Renin is enzyme. What is its function?
Oh,
Note down. I'll again describe this. The function of renin is to digest milk protein. The function of renin is to digest milk protein in young mammal. The function of renin is to digest milk protein in young mammal. Full stop. It is secreted in the young one, but secretion get stopped when they become young. So look, renin is an enzyme which is secreted in young mammals. So even in human, so to digest the milk, because the small kids, they are mostly or completely dependent on the milk for their nutrition. So milk digestion is very important part that takes place. So a special enzyme is secreted, that is renin. Now you are eating the cheese. Now many of you may stop the eating because, but nowadays there is a different process as well. But previously there were rennet tablets. Even still, some of them use it. Rennet tablets are made from the renin enzyme, and that renin enzyme is extracted from the stomach of calf, right? Stomach of calf, and this rennet tablet are used for converting milk protein into casein during the preparation of cheese and other milk protein, right? So during preparation of cheese, these rennet tablets are used, and rennet tablets are basically made from the renin enzyme, which is extracted from the calf stomach, right? Enzyme function outside of the living cell are called exogenous, right? So there are two type of them. The function which living, the enzyme which function outside of the living cell. So look, for example, uh, what is the function of tear, right? So tear gland, they are lacrimal gland, they are found here. Tears have enzyme known as the lysozyme. Lysozyme that is secreted along with the tear. When tear come out, lysozyme is secreted. That lysozyme kills the bacteria, right? So it is working externally, means it is working outside of the cell. So this is the lysozyme. And lysozyme, that is the IH2 uses. Enzymes which are functional inside the living cells are known as endoenzymes, right? Those enzymes which are working inside the cells, they are called endoenzymes, right? Enzymes, for example, those enzymes which are taking part in the Krebs cycle and glycolysis. So basically, Krebs cycle is the respiratory cycle that goes inside mitochondria, right? So those enzymes are basically of two types: exoenzyme and endoenzyme. So exoenzyme, they work outside of the cell, and endoenzyme, they work inside. Tell me, is it clear or not? Go through with it and let me know if you have any. All of you, please go through with it. Okay, clear. Martin, clear? Alia?
Yes, sir.
Fine. what is the economic importance of enzyme, right? So, like they are used as detergent. They contain proteases for brighter washing of the cloth and amylases for dishwashing. So proteases are a type of enzyme. They are used for the washing of cloth, and amylases are enzyme. They are used for washing of dish. Baby food. Trypsin is added in particularly pre-digested baby food. So those baby food like Cerelac and all that, right? The trypsin is added over here. So using baby food. The streptokinase. The enzyme it is used cleaning the blood clots. This question exclusively belongs to the medical examination, and number of the time I have seen this question. But now this is important for you as well. One marker question, right? So streptokinase is an enzyme that is used in cleaning the blood clots inside the blood vessel. So sometimes you must have listened that a person got blood clot, right? So this streptokinase enzyme is given an injection that it thicken the blood and it remove the clot inside the body. Digestive acid. Uh, Diastases and other enzymes are used in regularly by uh, uh, patient with a deficiency of digestive juices. So digestive acids are given to those patients where the secretion of digestive juices is less. Then lactase and lipase, they are employed to provide the proper uh, flavor to the cheese, right? And pectinase, it is used for cleaning the juices. And proteases, these enzymes are employed for chilling, uh, proofing of beverages, and giving the shining to the silk fiber, cleaning off the hides and all that, right? So these are the function of different, these are the importance of enzymes which are used in our daily life, economic importance. Now come to the chemical nature of enzyme. So here first, this is very important, very, very important. So normally enzymes are known, all the enzymes are globular protein except recently discovered RNA enzyme. So recently RNA is considered as enzyme also. So function of RNA is to forward the message of DNA or to carry the message of DNA. Along with that, sometimes RNA also work as enzyme, right? So recently it has been discovered that the function of RNA is also enzymatic, means it also sometimes work like enzyme. But except RNA, all enzymes are made up of globular protein. It is an enzyme which only made up of protein are called simple enzymes. So the enzymes which wholly made up of protein, they are called simple enzymes, like pepsin, trypsin, and urease. So the 100% they are made up of protein. So they are simple enzymes which are 100% made up of protein. Second category of enzymes, so enzymes are two types: simple enzyme and conjugate enzyme, right? So let me write it down. Enzymes are of two types. First, simple enzyme, and the second, conjugate enzyme, right? So simple enzyme and conjugate enzyme, there are two type of enzymes. So so simple enzyme are made up of 100% protein. The second category is conjugate enzyme, which are formed by two parts, right? They are made up of two parts: protein part called apoenzyme and non-protein part called cofactor, right? So now we are talking about we are not talking about the simple enzyme, we are talking about the conjugate enzyme, or they are also known as the holozyme. Try to understand. So simple enzyme are made up of 100% protein. There is no mixing of other things. So conjugate enzyme are also known as the holozyme. They are made up of two parts. Apozyme. So apozyme is proteinaceous part. We know all that, right? So this is basically made up of protein, right? And the second thing is cofactor. Cofactor is non-protein part, right? It is non-protein part, right? It may be, now the cofactor may be of two types, right? Either inorganic or organic cofactor. Inorganic cofactor, they include they include uh, normally minerals like calcium, mineral ions, right? They include mineral ions of uh, calcium, iron, copper, zinc, and etc. And if cofactor is organic cofactor, they may be of further two types, right? If it is organic cofactor, it may be of two types. That may be coenzyme or prosthetic group. Coenzyme, they are easily separable from non-protein organic part. Means this from non-protein easily separable from separable. They can be easily separated from the enzyme, and they are non-protein or origin, right? They are not made up of protein. Okay. Prosthetic group, they are non-protein and they remains attached to the enzyme, right? They firmly remains attached. So they firmly remains attached and they non-firmly remains attached, right? Like heme group. In hemoglobin, heme group is, that is prosthetic group. Okay. This is the diagram of enzyme where you can see this is inorganic cofactor, this coenzyme organic cofactor, this apozyme, and this part is called active site. What is the function of active site, right? Look, for sake of enzyme, a starch is digested with the help of amylase enzyme. Amylase convert starch into glucose, right? So this is amylase enzyme and this is a starch. Where it get fit, this starch molecule get fit, that part is called active site. At active site, this substrate means starch is substrate. So this substrate get fit, and after that it is broken down into glucose, right? That's how. So active site is that site, that functional site of the enzyme where actually substrate attached and the reaction start over there. Go through with it. Tell me if you have doubt, and if it is clear, you have to copy paste in your notebook right now as it is. First, ask me if you have any doubt. Then you have to note it down. Tell me. Siddhika, Martin, Elam, Alia. Okay, please note it down, and this is very, very important for those who also want to give NEET exam. Because in NEET exam, you won't get you'll hardly get question the rest of the part of the biomolecule part. You'll directly get the question from enzyme, and this is the most frequently asked part. So write down now, and let me know when you note it down. As it is, you have to copy paste it. And whenever you get the question about the enzyme, you have to draw this diagram. In the last test that I think taking place on the 6th of this month and 6 or 8 of this month, right? So only I got the copy of Siddhika only, and Siddhika, you have written very well, and your handwriting is very clear. Keep it up, right? And that was satisfactory uh, answer sheet, and the way she have written, it's very good, very lucid. The only thing which is missing with you, right, in some of the answers, not in all, some of the answers, I want more. I want you to make it more content rich, right? Uh, put the more and more information over here, right? Welcome. Put the more and more information over there. If you enrich it with the one thing, that is the put more information. Second thing, if possible, please draw a small diagram that will help you out. That will fetch a better marks, right? But anyway, you did a very good job, and I want that kind of the effort from every one of you. So first note it down, then I will show you that answer sheet of Chitika, and I want the same kind of the effort from Elam, Martin, and Alia as well, right?
Okay, sir.
Okay, fine, Shidika. Sometime I'll like put the diagram and I'll let you know that how to incorporate the diagram, right? In your answer. So first note it down and let me know when you note it down, when you copy it. And don't do take the screenshot and don't think that you'll write it down after the class. That is not going to happen. And many times you will miss all this content, right? Now the only 2 unit is left in your 2 unit are left in your syllabus, and by the way, they have more chapters. But plant physiology will do very quickly, and whatever the time that will take. So as per a scheduled time, as per I have committed with you, I'll finish the syllabus as per our commitment. In fact, before our commitment. Okay, Alia. Uh, fine. So I'll ask Sana to share with me so that I could check it, right?
Done, sir.
Done, Martin. Is that done?
Yeah, I mean, yeah,
Done or doing?
yeah,
Can you
done.
scroll down?
Okay, great. Okay, do it, Shidika. Okay. Also do I also write it on copy this diagram as well, the which I have drawn in the right side. Yeah. Okay, fine. Right. So okay. Now what is the active site? That is also a question. You may get the question, what is the active site of enzyme, right? I have made you understood that what is the active site. Let me read it out for you. The whole enzyme molecule is not active in catalyzing the chemical reaction. The only small portion of it is active. This is called active site. Enzyme may have several active sites. That is important. All the enzymes should end with a suffix "ase". Exceptions are some of the old enzymes. So nowadays in the modern enzyme nomenclature, we always add on suffix ase, like protease means digesting the protein, carbohydrate digesting carbohydrate, lipase digesting the lipids, right? So that kind of the RNAs digesting the DNAs digesting the DNA, right? So that's how. Carbo peptidase means digesting the carbohydrate and protein. Classification of enzyme. So that is the classification. According to older system, enzyme were classified into two broad categories: hydrolyzing and desmolizing. Hydrolyzing enzyme that catalyzing the hydrolysis of the larger molecule into a smaller molecule. Example, amylases. They often group into three categories. Proteolytic means breaking down the protein. Amylolytic, breaking down the carbohydrate. And lipolytic, digesting the or breaking down the lipids. So first hydrolyzing enzyme, then desmolizing enzyme. This is the older classification of the enzyme. Catalyzing the reaction other than hydrolysis, right? Aldoses, proteases, and catalyzers. The modern system of the classification was introduced by the IUB, International Union of Biochemistry, in 1961 into 6 categories, right? Oxidases. So first category. So as per the IUB, means International Union of for the Biochemistry, they have classified enzyme into 6 categories. Out of them, the first category is oxidoreductases. From heading itself or from name itself, you can understand what exactly they are doing. Oxidoreductases means they are oxidizing as well as reducing, right? So look, they takes part in oxidation and reduction reaction or transfer the electron, right? Here S plus S dash. So one compound is S and another is S dash. One will get reduced and another will be oxidized, right? In presence of oxidoreductases, right? And further, this S get oxidized and S dash get reduced, right? That is the function. They may be of three types: oxidases, dehydrogenases, and reductases. So oxidases, what exactly they do? They oxidize something. So like 2AH2. This is just a demonstrative reaction, right? It's not actual reaction. So here, what is happening? A is getting reduced, right? Sorry, A is getting oxidized. Look here, the hydrogen is getting removed from the A, so A will get oxidized, right? And will make a water. So they are called oxidizers. That's why enzymes are called oxidizers. The hydrogenases, they remove the hydrogen. AH2, hydrogen is removed. That is the hydrogenases. Reductases means they reduce, they add on the hydrogen. So A plus 2H, this hydrogen is added with the A, so they are reductases. Tell me, oxidoreductases clear or not?
Yes, sir.
Right? After the class, you have to copy paste it in your notebook. That's what I want. Yeah, sure, Shadika. I'll repeat it for you. My power backup exhausted, and it took some time to arrange me the secondary power backup. So actually, there was no light backup today. I'm extremely sorry for that. So I was here, right? I was repeating it again for Shidika. Tell me, try to understand. Look, so I have told you that there are 6 type of the enzyme, right? So there are 6 category of enzyme. Out of them 6 category, the first category is oxidoreductase, right? So oxidoreductases means it will oxidize, and either it will oxidize or it will reduce, right? So what is the oxidation? Oxidation is addition of oxygen molecule or removal of hydrogen, right? That is called oxidation. So you can understand here what will happen in oxidoreductases. There may be 3 types. So first type of oxidases means these enzyme will oxidize substrate. So here A is substrate. So 2AH2, right? This is a substrate. So A is substrate. Oxidases will add on oxygen to this A. So what is happening? Sorry, it will remove means just a minute. What is the oxidation? So oxidation, you can say A plus O. This is oxidation of A, or you can say there is a AH, removal of hydrogen, right? A plus H. This is also oxidation of A, right? Here you can see 2AH2. So A have hydrogen. Oxygen is there. Oxidases have removed hydrogen from the A. So oxidation of A means substrate taken place, right? And this hydrogen will meet with this O2 and it will make 2H2, means water molecule. So removal of water molecule have taken place. But A got oxidized, right? So that is the function of oxidases. Second one is substrate is AH2. Again, here A substrate, it is added with hydrogen molecules here, right? Now the second enzyme which is working over here, this is dehydrogenases. So what dehydrogenases will take place? Again, the same thing. It is removing hydrogen without oxygen, help of oxygen, right? So AH2, hydrogen have been removed. Again, the oxidation of A have taken place, right? Means oxidation of substrate have taken place. Third type is reductases. Reductases, so what is the reduction? So reduction we define like this way. Suppose that this is the AO. So removal of oxygen is called reduction. Or the second thing is addition of hydrogen is also called reduction. Here hydrogen is added on, so reduction taking place. So oxidoreductases may be of 3 types: oxidases, dehydrogenases, and reductases. Now tell me, is it clear or not? All of you, Alia, Martin, Shudika, Elam.
Yes, sir.
Clear? Shudika?
Yes,
Clear?
sir.
Great. The next one is that transferases. The function of transferases, they transfer a group from one molecule to another molecule, right? Group means functional group. They transfer a group from one molecule to another molecule. Glutamate pyruvate transferases transfer amino group from glutamate to pyruvate, right? The chemical group, look here, this S is substrate, and what is G? G is a functional group, right? Now it is reacting with S dash. Now what will happen? This G, this functional group will be transferred from S to S dash, right? Now transferases, what transferases is doing? Look here, they have transferred the functional group, they have removed the functional group, right? And they have transferred to someone else, right? So that is called transferases. They transfer the function. Clear or not?
Yes, sir.
The chemical group transfer does not occur in free state, right? Now hydrolases, they break, again every enzyme have ASES in their as their suffix. Here they break up large molecule to a smaller one with the help of hydrogen and hydroxyl group of water molecule. The phenomena is called hydrolysis, right? Try to understand. They break up the large molecule into smaller one. This is the function of hydrolysis with the help of hydrogen and hydroxyl group of water molecule, right? Look, H2O is a combination of two things: hydrogen group plus hydroxyl group, right? So their hydrogen is present, hydroxyl group. With the help of these two functional groups, they break down larger molecule into smaller one. These kind of the enzymes are called hydrolysis, right? So the phenomenon is called hydrolysis. This is also called hydrolysis, means breakdown of water molecule, right? So example are amylases, sucrases, and lactases. Here you can see maltose is C12H22O11, right? With the help of water, it breaks down into glucose, right? And those enzymes which break down maltose into glucose in presence of, means larger molecule into smaller one in help of water, they are called hydrolysis. Tell me guys, is it clear or not?
Yes, sir.
Very good. Liases. Try to understand. There are two kind of enzymes: ligases, liases. Ligases they add on, and liases they break down. Got it? Liases. So here we are studying about the liases, the enzyme that cause cleavage or removal of group without hydrolysis, means without help of water. Try to understand. It break down molecule, or it break down, remove the group without help of water, right? An addition of group to be double bond, or a removal of group producing double bond, right? Look here, C and C, these are the two carbon, they have different functional group. They break down these functional group right here, and they have broken down the functional group, and they have created here both of the carbon have single bond, but here the both of the carbon have double bond, right? So that is a function of liases. Here the water is not taking place. This reaction, which is given as an example reaction, it is a reaction of glycolysis. We will study this reaction in our chapter of respiration part, right? Fructose 1,6 biphosphate and in presence of aldose, so it break down into DHAP and G3. Go through with it and tell me the liase. Is that clear? Siddhika, Martin, Nalia.
Yes, sir.
Okay. Fifth enzyme is isomerases. They just make the isomers, right? The enzyme causes a rearrangement of molecule. Means what are the isomers? They have the same chemical formula but different arrangement or different structure. The enzyme causes rearrangement of molecular structure to effect isomeric changes. There are of three types: isomerases, epimerases, and mutases. Example: glucose 6-phosphate changes into fructose 6-phosphate. Formula is same but structure is different. Isomerases. Glucose 6-phosphate, glucose 1-phosphate, mutases. So just remember the isomerases. That's it. What are the function? They make the isomers. And you have to remember the first example. Again, ligases. Look, fourth one was liases. The sixth one is ligases. Where we have studied ligase? No, I haven't taught. Had I taught you ligases? No, I haven't taught you ligases. Ligases means ligation means to like to add on something, to join something, right? So ligases means they are synthesis. The enzyme catalyze the bonding of two chemical with the helping of energy obtained from ATP, right? Resulting in the formation of such bond: carbon oxygen bond, sulfur sulfur bond, carbon hydrogen bond, phosphorus oxygen bond. So by making the bond, what they do with the help of energy, right? They make the bonding. Like pyruvic acid plus carbon dioxide plus ATP plus water, they make the oxaloacetic acid plus ADP means energy is utilized, right? So that is the function of ligases. Tell me, is it clear or not? So there were six type of the enzyme. Now the next thing which now we are going to study, it is very important for your school exam. What they will ask you, if they as a long answer they can ask you, write down the factor affecting the enzyme. That is the first question. Or in 2-marker question, what is the impact of heat on the enzyme? What is the impact of concentration on the enzyme? What is the pH value, that and all that, right? So first, efficiency. The number of substrate molecule change per minute by a molecule or enzyme is called turnover number, or KET, right? KET. The higher the turnover number, the more efficient enzyme is. Clear? KET
Yes,
or
sir.
turnover number. Okay. So that is the turnover number. Next, heat sensitivity. Most enzymes operate optimally. This is very important. What is the impact of temperature? This is the most frequently asked question. What is the impact of temperature in the enzyme? So now try to understand what is the impact of temperature. So as the impact of any temperature, most of the enzyme they operate optimally, means normally with the full efficiency. Most of the enzyme they operate optimally between 25 to 35 degrees centigrade, right? So if a person get heat shock, means a person got heat shock or got too much heat, he or she died, because at the higher, when the temperature of body, internal body temperature increases, enzyme stop working and the person die. Sometimes people die because of the cold or frost. Even in the lower temperature, internal body temperature, enzyme stop working and a person or any organism die. So this is the reason that's why a person die in higher or lower temperature. The most enzyme operate optimally between 25 degrees centigrade to 35 degrees centigrade. They become inactive at freezing temperature and denature 50 to 55 degrees centigrade temperature. Means they become inactive at the below 0 degree temperature and below 50 or 55 degrees, above 50 and 55 degrees. However, the thermal algae or the bacteria, we have already studied that there are Thermus aquaticus bacteria, heliophytes, right, which can survive in the higher temperature. Their enzyme can work up to 80 degrees centigrade. The enzyme of seed and spores or also do not denature at 60 to 70 degrees centigrade, right? But these are the exceptions. So that is the impact of temperature on enzyme. Tell me, is that clear?
Yes, sir.
pH value. Each enzyme function at a particular pH value. This vary from enzyme to enzyme, right? Every enzyme have its own optimum pH value. On that pH value it works. Each enzyme function at a particular pH as pepsinogen at 2 pH, sucrose 4.5 pH, salivary amylase 6.8 pH. So every enzyme, salivary amylase stop its activity in the stomach where the hydrochloric acid is secreted, means where the pH is very low. Clear? Okay. Now the factor. So these were also the factor which we studied. The factor which affect the enzyme activity practically. The temperature at which enzyme shows the highest activity is called optimum temperature. It is generally correspond to the body temperature of warm-blooded animal. 37 degrees centigrade in human. Enzyme activity decreases below this temperature. High temperature destroy the enzyme by causing their denaturation. This occurs at 50 degrees centigrade. So in between the minimum or maximum temperature, the reaction velocity doubles for a rise of the 10 degrees centigrade. What does it mean? Look, the enzyme in our body, they work optimally at 37 degrees centigrade, right? So at rise of every 10 degrees centigrade, the functionality of enzyme get double. So the functioning of enzyme, that is at 35 degrees, right? 35 degrees centigrade. So when temperature from 35 to become 45, internal body temperature, the activity of enzyme become double, but it cannot go beyond 50 degrees centigrade. Beyond 50 degrees centigrade, enzyme will degrade and they will stop work. Did you got this point or not?
Yes, sir.
Yes, sir.
Great. Now concentration of enzyme. What will happen if concentration of enzyme increases? The rate of biochemical reaction rises with increase in the enzyme concentration up to a point that is called limiting or saturation point, right? So till saturation point, if the concentration of enzyme will increase, the rate of reaction will increase. Beyond this, increase in enzyme concentration have little effect. Means after a particular point, if you will increase the enzyme concentration, there will not be any impact or a very little impact. Product concentration. So enzyme, this is called like the starch convert into glucose, and amylase convert starch into glucose. So starch is substrate and amylase product. So if product concentration means the concentration of glucose will increase, then what will happen? The rate of forward reaction will fall. Reverse reaction can also start, right? If the product concentration will increase, the rate of reaction or rate of reaction of enzyme will decrease. What is protein poison? Cyanide, azide, means they stop the activity of enzyme, right? What will happen with the substrate concentration? Increasing in the substrate concentration, increase the rate of reaction of enzyme, right? Just this Michaelis constant, it is not required for you. Activation energy, you can understand. This is in your NCERT. So, what is the? Can you tell me what is the benefit of enzyme? Any one of you? What is the benefit of it? How it is usable for our body? Yes, Martin. Jyotika? Enzyme help in multiple factors of the body. Yeah, they function, but how they are functioning? Look, suppose that there is a reaction which can take place without enzyme, or which can take place with the help of enzyme. Right? Means that is the reaction that may take place without enzyme, but that will also take place with the help of. Suppose that I eat a bread, means a starch. So to digest that 100 gram or the 1 gram starch, it requires 300 kilocalorie energy to complete the step. But if you will if you will put enzyme in that starch, in place of 300 kilocalorie energy, it will require only 10 kilocalorie energy, right? So that reaction or rate of reaction is increased, right? And the energy level gets down. At the lower energy, that reaction can take place. Did you got that point or not? Martin, are you getting it or not? Tell me.
Yeah, yes. Transition state. Can you explain the activation?
Yeah. That I'm going to. Just I was telling you with the help of like a layman language. So look, here I have given example. What is the function of enzyme? Protein break down into amino acids. If this reaction is taking place without enzyme, it will take 300 kilocalorie energy. But when we put enzyme proteases, the same reaction takes place in 25 to 30 kilocalorie. Right. So look, this is a diagram. This diagram you have to draw. If they ask you the question from the activation energy, this is very, very important question. They can ask you what is the activation energy. If they ask you what is the activation energy, you have to draw this diagram as it is. Look, this is the energy potential, and this is the progress of reaction. Normally, a substrate convert into product, right? And their transition of substrate into product, it requires some energy. The energy which is required for converting any substrate into product, that is called transition state or transition energy, right? Or activation energy, right? So activation energy without enzyme is very high. But when you will add on the enzyme, the activation energy become low, right? That is the functioning of enzyme. That's what this graph explains, right? If you will add on the enzyme, the activation energy will decrease and transition state will take place at the lower energy level, right? If you are not adding the enzyme, the activation energy will be very high for the same level. So without enzyme, body will take too much energy to digest food. But with the help of enzyme, it is taking too little energy to digest it. Alia, tell me. Did you got it? What I say? Elam, tell me. Is that clear, Elam?
Yes, sir. Yes, sir.
Okay. So what is the activation energy? An external supply of energy is needed for the start of a chemical reaction. It is called activation energy. For example, acidic hydrolysis of sucrose require 32,000 kilocalorie calorie per mole energy. This is large amount of energy cannot be provided by living system. Enzyme lower the activation energy required by the reaction. This is achieved by four ways. Dissolution or taking a reactant out of solution state, establishing weak bond between the reactant and enzyme. It releases the energy called bond energy, bringing the uh, bringing the reactant molecule closer to each other, development of strain in bonds, formation of. By the way, this is not required for you if you keep it till this point. If you can include this part, that is very good. But even if you don't include that part, that is also okay. Tell me, activation energy, did you got it or not? All of you, I want
Yes,
consent
sir.
from all
Yes.
of you.
Yes, sir.
Very good. Last but not the least, how does enzyme work? You have to remember this first one, right? This is called lock and key model. Like every lock have its own specific key. The same way, every substrate for digestion, it have a particular enzyme. So enzyme is key, right? And the substrate is lock. Here, this diagram or this model was represented by Emil Fischer. They said that activation, the shape of activation, active site matches with the shape of substrate, right? And the enzyme and substrate, they make the complex, right? And then enzyme, the substrate become product, and enzyme is reutilized, right? That is the called key and lock model, right? So like every lock have its own key. In the same way, every enzyme have its own specific substrate. Is that clear? So this was all about the biomolecule part, right? And for this chapter, go through with your NCRT and ask me if you have any doubt. And you have to note it down, these notes. You can download it from here. I have attached the file as well from here. Look, this file is attached over here. Here, from here you can download it, right? And the file is attached. And I'll say that you need to note it down from there. This is my notes, right? And go through with the NCRT and ask me if you have any doubt. So we have completed the cell biology completely today. In the next class onward, we will start the plant physiology part, right? So tell me if you have any doubt. Ask me. Okay, okay, Shritika, fine. So Martin, Alia, all of you, please tell me if you have any doubt.